Hi everyone, and you're very welcome to today's Saturday session, which is going to focus on Leaving Cert Biology, and our topic for today is plant structure and plant transport. So the first thing we're going to look at is the general structure of flowering plants, and plants can be divided into two portions. We have an overground root system and we have an underground root system, and we can see that in the diagram over here on the right hand side. So here we have our overground shoot system, so anything above the soil, and our root system is anything below the surface of the soil. So we have our roots. In the shoot system up above, we generally have a flower, we have apical buds, we have nodes, internodes, apical buds, vegetative shoots, we have leaves, which are composed of blades and petioles, we have axillary buds, and we also have our stem. And then we can have different types of roots, which we will get to in a little while. So this one here is a taproot. We also can have fibrous roots as well. So that's the general structure of a flowering plant, and we're going to look at a couple of functions of each individual part. So the roots then, the function of the root is to anchor the plant in the soil, okay? A little bit of support for the plant to make sure that it remains upright and isn't impacted by any natural weather like wind. So the roots also absorb water by osmosis. So again, just really quickly, what osmosis is, there we have our semi-permeable membrane. And over here on the left-hand side, we have a big amount of water molecules. On the right-hand side, we have a small amount of water molecules. So if we were to imagine this was the soil and this was the internal root structure, the concentration of water is much higher on the outside of the root than it is on the inside of the root. Now, osmosis is a passive process, there's no energy required, and the water is going to move from a region of high concentration to a region of low concentration so that the water balances out. And this is what happens if we were to water a plant. There will be a huge amount of water concentrated on the outside of the root, so it's going to move by osmosis into the root. Now, minerals will also move across into the root as well. It's not just water, minerals as well. These absorbed minerals will be transported by the root to the shoot, and roots can also be used for food storage as well, which we will get to in a little while. Now, we mentioned a couple of seconds ago about how we have different root types. So two root types which we have here are our taproot and our fibrous root. So the taproot, we can see here, has a much larger structure here towards the centre, where the fibrous root, it just looks like loads of little fibres. So the taproot is referred to as a primary root, and it consists of one main root growing from a radicle. The fibrous root has equal sized roots emerging from the base of the stem. So we can see here, here's our primary root, that's the taproot, whereas the fibrous root has equal sized roots all emerging from the base of the stem. So that's our two different types. Now we're going to look at a longitudinal section of a root. So there's two types of cuts we could make if we were looking at a root or a shoot. We could have a longitudinal, or we could have a transverse. Now the longitudinal will be cut down, the transverse will be cut across. So you can imagine if we were looking inside into a root, we sliced right down through it, this is what we're going to see. So we have our xylem, which is responsible for transporting water and minerals. And we have our phloem, so an F sound in phloem, an F sound for food, so that's going to transport food. We have our root hairs, which we will come to in a couple of minutes. We have our epidermis, which is used for protection. And then we have a couple of other regions here. So we have our differentiation region, we have an elongation region, we have a cell production region, and we also have this meristem region. So just quickly to mention here, we will come back to this in a couple of minutes. This is where cell division occurs rapidly, okay, so in the meristem. Here we have our root cap, which is used for protection. So it's at the very, very tip of the root going down into the ground. So that's our root cap. Now we can see here our xylem is towards the centre, the phloem is towards the outside. That's important to know when we come back to that in a couple of minutes and maybe look at the transverse section to figure out which one is which. Now we mentioned these zones already, and I will add the fourth one in a second, which was the meristem, which we mentioned a couple of minutes ago. But the first zone we're going to look at here is the zone of protection. And that does exactly what it says in the name. It protects the root as it pushes through the soil, okay? And that's what the root cap is responsible for there. The zone of cell production, also known as the meristematic region, that's which allows the plant to grow. So there's rapidly being new cells produced by mitosis. So this is where one cell becomes two identical copies. It's in the meristematic zone. So this is where growth occurs. So most of the growth is going to occur in that region down there. So the meristem, rapid growth of cells there. And we also have our zone of elongation. So again, does exactly what it says in the name. This is where cells increase in size, and this allows growth to occur. And this can be impacted by growth regulators. So an example, auxin, which is a growth promoter. So it's going to allow the particular plant, or the root in this case, to grow. So that's the zone of elongation. Now we're just going to mention another zone here known as the zone of differentiation. And this is really important for these elongated cells which are beginning to mature and develop. So these elongated cells can develop into different tissue types within the plant. We can have dermal tissue, we can have vascular tissue, and we can have ground tissue. Now the dermal tissue, think of your skin, think of the epidermis, that's generally used for protection. The vascular tissue, that's your xylem and your phloem. So that's responsible for transporting food and water and minerals. And then we have our ground tissue, which is generally found just between the dermal and the vascular. Okay, so dermal towards the outside, vascular towards the inside, and then ground tissue towards the center. So when the cells are beginning to mature, it can turn into one of three different tissue types: dermal, vascular, or ground. So this is the two different sections, or the two different cuts which you can make in a root, and this is going to show you the location of the xylem, the phloem, the epidermis, the ground tissue, and the vascular tissue. So if we look at the longitudinal one first of all, which is the one we had a look at a couple of minutes ago, we can see here we have our xylem towards the center and our phloem towards the outside. And that's exactly what we can see over here as well. So the xylem, letter X for xylem, letter X here in the center for our xylem. Again, we can see that's towards the center, whereas the phloem is what surrounds that. So that's more towards the outside. So the vascular tissue, xylem and phloem, is towards the center, which is exactly what we said a couple of minutes ago. Now on the outside, this is where we have our dermal tissue. So this is the protective one. So we can see epidermis there on the outside. That's used for protection. And we can see that's exactly where it is over here as well. In the transverse section, the epidermis is towards the outside. And the ground tissue is located between both of these. So we can see it over here on the right hand side, and that's what will be the tissue here over on the left hand side as well. And just to mention again, the root cap, this is where it's pushed down into the soil. That's going to provide protection for the plant as it's going down into the soil. And the meristem, rapid cell production there. And then the cells will start to elongate in the zone of elongation. So the stem then, we can break this down into two different types. We can have a herbaceous stem or a woody stem. Okay, so herbaceous plants and woody plants. So herbaceous plants do not contain wood, whereas woody plants, it's in the name, they do contain wood or lignin. So an example of a herbaceous plant would be a daffodil. An oak tree will be an example of a woody plant. So the function of the stem is to support the aerial parts of the plant. So the root is going to provide support for the entire plant. The stem is going to support the aerial parts, so any of it that's in the air. It's also going to transport water and minerals from the roots to the leaves. So any of the water and minerals that are after being absorbed by osmosis is going to move from the water up to the leaves through the stem. But it's also going to transport food from the leaf down to the root. Now, why does that make sense that it's moving that way? Well, photosynthesis is the process by which plants make their own food using sunlight. That happens in the leaf. It's not just the leaf that needs food, the entire plant needs food, so it's going to have to move from the top of the plant right down to the bottom towards the roots. And some stems can also be responsible for storing food there as well. Now, different parts that the stem have, we have our apical bud. This is the tip of the stem, allowing the stem to grow. Okay, so it's kind of like the zone of elongation that we had over in the root. So this is the apical bud up here. It's going to allow the stem to grow. We have a node. So this is the point on the stem where leaf is attached. So we can see the nodes here. Leaves will grow out of that. We have internodes, so inter meaning between. So internodes is between nodes. That's just any region between two nodes. So we might have a node there, we might have a node down here. So this entire region here is known as the internode. The bud is a potential growth point on the stem, which may turn into a leaf or a flower. Okay, so we can see lots of buds over there on the right hand side. We have our lenticel, which are tiny openings here. We can see there's lots of them along the whole stem. They're openings for gas exchange. So gas exchange doesn't just happen in the leaves, it can happen along the stem as well. And then we can have some scales and scars. So these mark the location of previous apical buds. So they obviously, as it's growing, can be removed or lost. So they are some scars that are left over. So we're going to look for similar tissues that we had in the root. So we're going to look for our dermal tissue, we're going to look for our ground tissue, and we're going to look for our vascular tissue. So again, on the outside, this is where we're going to have our dermal tissue, so the epidermis, which is going to provide protection for the plant. Towards the inside, we can see here we have our vascular bundles, so it's a little bit different from the root. They exist in what we call bundles. But again, if we have a look, the xylem is going to be towards the inside all of the time. It's always pointing towards the inside, whereas the phloem is always pointing towards the outside. So very similar to the root. Now our ground tissue, again, like in the root, that's what exists between the dermal tissue and the vascular tissue. So one thing just to keep an eye on in here is that those vascular bundles, they're scattered around. Okay, so there's no particular order to where they are. That actually tells us about the type of plant that it is, because it's either going to be scattered around or they're going to be on a ring on the outside. So because they're scattered, it's a particular type of plant, which we'll come to in a couple of minutes, but it's just something to keep an eye on for now. But again, we have the exact same tissues here. We've our dermal, we've our ground, and our vascular, pretty much the same locations. Dermal towards the outside, vascular towards the inside, and ground in the space in between. Now we're going to have a look at the longitudinal section as well, so very similar to the transverse section. It's going to be the same tissues in pretty much the same locations. The dermal tissue is going to be towards the outside, the vascular tissue is going to be towards the inside, and we have the ground tissue, which is in between. Again, the exact same idea here. We have our xylem tissue is towards the inside, and we have our phloem tissue is towards the outside. So just remember, xylem is always towards the center, phloem is always towards the outside. Now we're going to focus in on the leaf for the next couple of minutes. So I mentioned a couple of minutes ago about how food is transported from the leaves down through the stem towards the root. And the reason that it's transported from the leaves down is because photosynthesis occurs there. And photosynthesis is the process by which green plants make their own food. Okay, so leaves are responsible for food production. They're also responsible for transpiration, so this is where water is lost out of the leaves. They're responsible also for gas exchange. Okay, so carbon dioxide is taken in, oxygen is released. We then in turn can breathe in that oxygen. And again, just like the root, just like the stem, they can also be responsible for storing some food. So these can have different patterns of veins then, and it's these veins that contain the vascular tissue, which is the xylem and the phloem, which we've mentioned a couple of times already. So the two types we can have parallel veins, so it looks like we're forming parallel lines down here, or they can be more netted. So we've already mentioned there about the tissues in flowering plants. Many times we've mentioned about the dermal tissue, the ground tissue, and the vascular tissue. So the dermal tissue is more towards the outside. The vascular tissue is more towards the inside. That's the xylem and the phloem. And the ground tissue surrounds. Okay, so surrounds the vascular tissue. It's in between the vascular tissue and the dermal tissue. So the meristematic tissue, think of the meristem. Okay, so rapid cell production. So within that meristem, many cells are going to be produced, it's going to divide, and it's going to differentiate into the three types of tissue. So it's either going to become dermal tissue, it's going to become ground tissue, or it's going to become vascular tissue. So again, just to go over all of the tissue types, because they're going to be really, really important for us. We have our dermal tissue, which is the covering layer used for protection, and the epidermis of leaves and the stem is coated with a waxy cuticle. And we can see that down in the diagram here. You could even try this at home. If you went out into your garden and you took a leaf off one of the plants that you would have there, you'd notice that the upper layer is more waxy in look than it is for the bottom. So if you put that leaf under the tap for a couple of minutes, instead of the water actually sticking to it, it will form these beads and actually run off. So it just means that if the plant was in a location where there was heavy rainfall, it's not going to be weighed down. The water will all run off the leaf, preventing it from being damaged. The ground tissue, this is the tissue between the vascular and the dermal tissue. Some of the functions include photosynthesis, storage of food, storage of waste, and provide strength and support to the plant as well. And we can see it in the simplified diagram down here. We have our vascular tissue towards the centre, again xylem at the very centre, phloem surrounding that. We have our dermal tissue on the outside, and it's the ground tissue that exists in between, offering a little bit of strength and support. And then our vascular tissue, this consists of xylem and of phloem. So the primary function is to transport materials throughout the plant. Xylem responsible for the transport of water and minerals, phloem responsible for the transport of food. And it doesn't matter whether you're looking at the longitudinal section or the transverse section, the xylem is going to be always towards the centre. The phloem is always going to be towards the outside. Okay, really, really important. And you can kind of see that over here when you look at the transverse section here. It's kind of more like the X shape in the root compared to the stem. So this is it in the root, this is it in the stem. And this actually hits on the point we made a couple of minutes ago about the arrangement of the vascular bundles in the stem. I mentioned a couple of minutes ago that because these particular bundles were scattered the whole way across the stem, that told us about one particular type of plant it was. This is arranged in a ring, which tells us the other type of plant it is. And again, we'll come to all of that in a couple of minutes. So we're going to focus in now on the structure of xylem and phloem for a couple of minutes. Now, the xylem consists of two types of cells. We have vessel elements and we have tracheids. Okay, we're going to focus in on the vessel elements first of all, which are these structures down here. So the first thing we're going to look at is the overall structure and how they kind of resemble tubes, but that they have these holes on the outside. We'll come back to those now in a second. So these tubular structures are joined end to end. So we can see this is where they would join here and here. So they're joined end to end and they can form continuous tubes. They're much wider than the tracheids, and we can see that from the diagram as well, and they're much more efficient in transporting water. Now these cell walls contain lignin, and this lignin provides some strength. Okay, so the cell walls contain lignin, they provide strength. The tracheids then on the other hand, they're long sloping tapering cells, and what we mean by tapering is they're kind of cut at an angle up here at the top and the bottom, and they overlap to allow water to pass from cell to cell through thin sections called pits. So rather than the water moving up and down, most likely in the vessel elements, the water is actually going to move side to side in the tracheids. And one thing to keep in mind here is that these xylem cells are actually all dead cells. So they're not living, they're dead cells. So just keep that in mind for later on. So we have our vessel elements and our tracheids then for xylem. Phloem on the other hand, we have our sieve tubes, sieve plates and companion cells. So if we have a look here, these companion cells are towards the outside. We're going to look at the sieve tubes and sieve plates as one whole now. So the sieve tubes, these are long tubular structures, kind of like the vessel elements from before. We don't have the holes at the side. So we have our long tubular structures, so there's one there, and they form when cells join end to end. Okay, we can see that we do have some gaps in there in the centre as well, and these come from our sieve plates. So these sieve plates develop pores to allow the passage of materials from one of these sieve tubes to another. Now each sieve tube, if we look to the side of it over here, has what we call a companion cell. And this contains a nucleus and a cytoplasm. So the nucleus is this structure here, the cytoplasm is this jelly-like substance that surrounds it. And that controls the sieve tube activities. Okay, so the nucleus, if we think of the brain of the cell, it controls cell activities, that's going to control what this sieve tube does. Now the primary function of the phloem is to transport food from photosynthesis to leaves, downwards usually, but sometimes upwards. So photosynthesis is the process by which green plants make their own food, generally occurs in the leaves. It's not just the leaves that need the food, the whole plant needs the food, so the food is going to move down. Sometimes it can be up as well, but generally it's going to be down. Now, the xylem structures on the other hand are going to move upwards. So the water is going to generally move up, the food is going to move down. And one big difference between the phloem and the xylem, we mentioned already about how the xylem is dead cells, the phloem are living cells, okay? Has our nucleus, so they're going to be living cells. Now, a couple of differences here. Xylem and phloem. So xylem carries water and minerals, F sound in phloem, F sound in food. Xylem is dead, phloem is living. Xylem has lignin, phloem does not have lignin, and the xylem does not have companion cells, where the phloem does have companion cells. So a couple of differences there between our xylem and our phloem. Now, I mentioned a couple of minutes ago already about how the vascular bundles can either be scattered throughout the stem of a plant or they can be arranged in a ring, and this is leading to this here. So plants can be divided into two categories. They can be monocotyledons or monocots, or they can be dicotyledons or dicots, okay? And this is where a cotyledon is a seed leaf. So do they have one seed leaf or do they have two seed leaves? So mono meaning one, di meaning two. Now, you can tell if a plant is a monocot or a dicot by looking at a couple of its features. So if you examine the seeds, the roots, the vascular bundles, the leaves and the flowers, you generally can tell straight away whether that plant is a monocot or a dicot. So we're going to focus here on the vascular bundles first of all. So the first one we looked at today was where they were scattered throughout the stem. If they're scattered, that means it's a monocot. If they're arranged in a ring around the outside, that means they're a dicot. A couple of other features: if there's only one seed leaf, obviously that means it's a monocot, because mono meaning one. If it has two cotyledons, that means it's a dicot. Monocots in general will have fibrous roots. Dicots would normally have taproots. And we've already mentioned about the vascular bundles there. In monocots, the leaves would generally have parallel veins, whereas in dicots it would be net-like veins. And then flowers as well. So monocots would generally have number of flowers in multiples of 3, whereas dicots would be multiples of 4 or 5. So an example of a monocot here would be something like grass, and an example of a dicot would be any legumes. So for example, the pea plant. So we're going to have a quick look at an exam question here. So it says, draw a diagram of a transverse section through a young dicotyledonous stem as seen under the low power lens of a microscope. Indicate on your diagram a location for each of the following: dermal tissue, ground tissue and vascular tissue. Now, really important here to look at the keywords. So it says a dicot. So we've known from the last slide there that the dicot is going to have the vascular bundles arranged in a ring. So we're going to have transverse section as well. We're cutting across. So we're going to have our dermal tissue on the outside. We're going to have our vascular bundles arranged in a ring on the inside. Again, split in two. I'm just going to color coordinate it just to make it stand out a bit. So the phloem is going to be towards the outside. The xylem is going to be towards the inside. Collectively, they're known as the vascular tissue. And then the ground tissue is all of the tissue in between. So you can color that in, whatever. That's the ground tissue. Okay, so that's all we needed for that one. Which of the above tissue types has a different location in a young root? So this one is going to be the vascular tissue. And the reason being is because it's found in the center of the root. When we look at the transverse section of a root, that was the next question there. Where precisely is this? It's more towards the center. It's not in a ring towards the outside. So give one function of the ground tissue. One of them is just support. It can obviously store food, and then photosynthesis will be another one there as well. Draw label diagrams to show the detailed structure of the two vascular tissues of plants. I'm not actually going to draw them, but that's the tracheids, vessel elements, and then the sieve tubes. But I will do the next ones here. Which of the tissues referred to in part 3 is composed of living cells? That's the phloem, because xylem is dead cells. And what is the function of meristematic tissue? So this is for cell division, so to produce more cells. It can also be for growth there as well, but you actually only need one function there. Okay, so that's an example of a question which has come up on the structure of plants. Another one here. So this diagram shows the transverse section through the stem of a monocotyledonous plant. Again, makes sense because all of the vascular bundles are spread around. What is the term? What's meant by the term monocotyledonous? So it contains one seed leaf. Give an example of a monocotyledonous plant. So one of them would be grass. Name the structures labelled A. They're the vascular bundles. How do you know from the diagram that the section is taken from a stem? Well, there's more than one vascular bundle. And how do we know it's from a monocot? Scattered vascular bundles. How are the veins arranged in the leaves of monocots? So if you think back to the grass being an example, we have parallel veins. We mentioned that a couple of minutes ago. And how does the vein arrangement in the leaves of dicot plants differ from that in the monocots? So it's more branched in dicots. So it's not parallel, it's more branched. Okay, so for the next couple of minutes, we're going to look at plant nutrition, which is the way in which an organism gets its food. But more particularly, we're going to look at the way in which plants get their own food. So there's two different types. We can have autotrophs or we can have heterotrophs. Okay, this applies to all organisms. So the word auto refers to self, troph referring to food level, and the word hetero means different. So an autotroph makes their own food, whereas a heterotroph gets their food from other sources or from their environment. So we will be an example of a heterotroph because we get our food from other sources, whereas plants will be an example of an autotroph because they actually make their own food in a process known as photosynthesis. And the food source that they make is referred to as glucose. So we've mentioned already about how plants are autotrophic. They make their own food through photosynthesis, but they need to be able to take in and transport water, carbon dioxide, oxygen, and other minerals. These are all requirements for plants to be able to make their own food. So if we look at the equation for photosynthesis, it's carbon dioxide and water. Sunlight is trapped by chlorophyll, and we get glucose and oxygen. Okay, so for this to happen, the plant needs to be able to take in carbon dioxide, needs to be able to take in water, needs to be able to trap the sunlight, and it needs to then be able to transport the glucose around its entire structure and also to release the oxygen. Okay, and we're going to look at each one of these individually as to how it's taken in and possibly how it's transported or released. So a couple of things just in terms of the light. This comes from the sun and is trapped by the chlorophyll within the leaves. The water enters the roots through osmosis and travels to the leaves through the xylem vessels, which we'll come to again in a couple of minutes. The chlorophyll is the green dye in the chloroplast. That's what's responsible for trapping the sunlight. And the carbon dioxide, that comes from the air. Obviously, we breathe it out. That's one of the reasons or one of the places it comes from, and it enters through these tiny holes called stomata on the underside of the leaf. Now, the first thing we're going to look at here is the uptake of water. So how does water actually get from the soil into the plant? So it's going to happen by osmosis. And again, we've already looked at this, but if we have our semi-permeable membrane, we have lots of water molecules on one side of that semi-permeable membrane and very few on the other. Osmosis is the movement of water molecules from a region of high concentration to a region of low concentration. It's a passive process, no energy is required. That's exactly what happens for the uptake of water in the root. So it's absorbed by the root hairs, so it's these structures that are extended from the main root. And because there is such a large number of these root hairs, it allows for the maximal absorption of water. So absorption, as we've said, occurs by osmosis. And we can see that happening over here. We have a huge amount of water molecules outside of the root hairs. We have very few of them on the inside. So once water comes in contact with the root hairs, it's going to move from a region of high concentration to a region of low concentration, passively, no energy required, through osmosis. Now, how does the water actually move from the roots the whole way up through the plant? So it moves up through the plant as a result of two factors. We have our root pressure and we have transpiration. So if we have a look over here, this was a simple experiment done to represent root pressure. So at the beginning of the experiment, we had a certain amount of water here. This is representing the root, and it was cut off down here. Okay, the water level was marked off. This is a glass tube just showing where the level of water was in each case, and this here was the root. Okay, this was a stump of a plant connected to the glass tube. So more water would have actually been added into the soil, and the pressure that was forcing this water up, you can actually see cause the water level to rise. So the pressure that's being exerted down here in the soil is actually pushing the water, not hugely, but it is pushing the water up through the root and into the beginning of the stem of the plant. Okay, and we can see the raise there in the water level. The other factor that's responsible for getting the water from the roots up to the leaves of the plant is transpiration, and this is the upward pull of water through the plant. So transpiration is the loss of water through the surface of the leaves. The sun obviously helps with this, but it's an upward pull, so the water is essentially being pulled up through the stem. Now, how does that actually make sense, that this water can actually be pushed up and stay up, but then transpiration can continue to pull the water from the roots up to the leaves without it going backways or there being a break in the water? Because we can see here, this is a continuous flow of water. There's no gaps at all. That's because of two things. So these two things are cohesion and tension. So this was actually come up with, it was first proposed by two Irish scientists, Henry Dixon and John Joly, and they came up with the cohesion-tension model, sometimes known as the Dixon-Joly model. Now, the two words we're going to focus in on here are cohesion and tension. Okay, we're also going to focus in on adhesion. So cohesion, so if something is it's similar. This is the sticking of similar molecules to each other. Essentially what's happening here for cohesion is the water molecules are sticking together. That's why there was no break in the flow of water. That's why there was no gaps in the water in the previous slide. There's cohesion there, so the water molecules are sticking together. Adhesion is the sticking of different molecules together. So down here, an example of that will be the water and possibly the outside of the stem of the plant. So the water molecules are sticking to the cells that are in the stem of the plant, meaning that when they move up, they're not falling back down because they're sticking to the outside of the plant or sticking to the lining of the stem. Okay, so that's cohesion and adhesion. Cohesion is the sticking of similar molecules. Adhesion is the sticking of different molecules together. Okay, so now we're going to look at the minerals and the uptake and transport of minerals within the plant. And plants require a variety of minerals. Again, these are going to be transported within the xylem along with the water, because the xylem is responsible for water and minerals. And some common minerals that a plant would need include calcium and magnesium. Now, these are always going to be dissolved in the soil and they're going to be absorbed by the roots. Not too dissimilar to the water being absorbed into the roots, but it is actually a different process that happens here, which we'll get to in a minute. So this process to get those minerals from the soil into the roots, it does require a small amount of energy. So it's not like osmosis, which was the movement of the water molecules from the high region to the low concentrated region. It was a passive process. There was no energy required for the water to move. There is energy required for these minerals to move. And this process is known as active transport. Okay, so this is the movement of particles across a semi-permeable membrane, okay, but energy is required. So active transport, energy is needed. Once these are moved across the membrane, they are able to be transported along with the water in the xylem, okay? But the big difference there, just remember this between the minerals and the water. Water, no energy is required. Minerals, there is a small amount of energy required. That's why water moves by osmosis and then the minerals move by active transport. Okay, so the one we're going to be focusing on here now is glucose. So this is the transport of food. Now we've mentioned already about how glucose is produced by the plant during photosynthesis. It's produced in the leaves. Okay, this is then transported in the form of sucrose from the leaves down through the plants. So we've mentioned this already, how water moves from the roots up to the leaves, whereas the food moves most of the time from the leaves down towards the roots. Okay, so it's an opposite movement there. And this is done by the phloem. So just remember F sound for phloem, F sound for food, and the food in this particular case is the sugar. Now the final thing we need to look at here is the idea of carbon dioxide and how this is actually taken into the plant. Because remember, the formula for photosynthesis is carbon dioxide and water. Sunlight is trapped by the chlorophyll to give glucose and oxygen. So we've discussed all of those so far apart from the carbon dioxide. How do we get this into the plant so that photosynthesis can occur and the plant can make its own food? So glucose is produced by plants during photosynthesis, but one of these requirements is carbon dioxide. The majority of this carbon dioxide is taken in through the stomata in the leaves by diffusion. Okay, so if we were to get a leaf sample and look at it side on, we would see right here at the bottom will be an opening. Okay, these are referred to as stomata. They're tiny, tiny little holes, and this is what allows the carbon dioxide to get into the leaf. Okay, we can see here we do have the dermal tissue on the outside, which is going to be for protection, but we do have these extra structures here called guard cells. And these guard cells basically open and close, cause these stomata to open and close. They control the opening and closing of these stomata, and that allows carbon dioxide to get in, but also allows for some oxygen to escape as well. Okay, so stomata are the tiny holes at the underside of the leaf, and that's where the carbon dioxide will enter. Now, how does this gas exchange actually happen? Well, first of all, before we even look at the text, we're going to look at the diagram over here on the right-hand side. So during the day, the stomata are open, so the plants are actively taking in carbon dioxide. Again, that makes sense because they need sunlight for photosynthesis to occur. Majority of the sunlight, if not all of the sunlight, is going to happen during the daytime. Okay, so the stomata are open, allowing carbon dioxide to come in. This means the guard cells are swollen or turgid. Okay, so they swell up, causing this opening. At night time, though, the stomata is closed. The guard cells have shrunken, they're flaccid, so very little, if any, carbon dioxide is going to be able to get into the plant. Okay, so we can see the big difference here between daytime and nighttime. Now we go through that in a little bit more detail. So each stoma is enclosed by a pair of guard cells. So these are our guard cells here. They open and close by changing shape. So if they're swollen, they're open. If they're shrunken, they're closed. High levels of CO2 will cause the stomata to close, and low levels will cause them to open. So oxygen will diffuse out and CO2 will diffuse in. Now, what do we mean by diffusion? Very, very similar to osmosis. So it's a passive process. It's the movement of molecules from a region of high concentration to a region of low concentration across the semi-permeable membrane. So osmosis, I suppose, is a special type of diffusion involving water. Diffusion happens with all other molecules. So oxygen is going to diffuse out. That's a byproduct of photosynthesis. CO2 is going to diffuse in, okay, because that's going to be used for photosynthesis. So we're going to have a look at our last exam question here. So plants require water for survival. By what process does water enter the root hairs? So this particular process here is osmosis. Part 2, draw a large diagram of a transverse section of a root and label the following tissues. So the transverse section of the root is going to look something like this. The xylem is the X shape. The phloem is on the outside. Altogether, that's the vascular tissue. We have ground tissue in the middle. We have our dermal tissue towards the outside. And don't forget, we do have some root hairs there as well. Okay, so that was part 2. Part 3. Name the 2 Irish scientists who first described the upward movement of water in plants. So you don't need their first names here. It's Dixon and Joly, but we'll give them their first names here. It's John Joly and Henry Dixon. And then part 4, describe in detail the upward movement of plants. We actually needed 3 points here. We want to try and use adhesion and cohesion. So we have root pressure as well. So that pushes water slightly up the stem. Transpiration pulls water up. Adhesion sticks water together. Cohesion sticks water molecules together. Adhesion prevents, I'm actually going to have to do this up here. Adhesion prevents backflow. The backflow of water by sticking water molecules to the xylem, or the walls of the xylem, and then the water moves up. Okay, so lots of points there. You could mention there's a little bit of hydrogen bonding as well, and it is a continuous column. There's lots and lots that you could put in for that particular question. Okay, so that's pretty much it for plant structure and function today. We've looked at the structure of the root and the shoot. We've looked at the longitudinal section, the transverse sections. We've looked at the difference there between monocots and dicots. We've looked at how carbon dioxide, water, food, minerals all enter the plant and how they're moved. And we've had a look at a lot of different exam questions throughout as well. Now, before we go, I'm actually going to show you around the ExamRevision website where you can access lots of more content like what we looked at today. So we'll do that on the next slide. So just to finish off, I'm just going to quickly show you around the ExamRevision website. So you can see here for Leaving Cert Biology and for all of our subjects, we have video tutorials, quizzes, presentations, an exam builder and a resource pack. Now the video tutorials, if we want to find the videos associated with today's content, it's in 3.2. You can see there we have a whole host of videos. So you can click into those and watch them again and again at your own pace, covering the entire content. Once you've watched the video, you'll be able to click in and do an MCQ self-correcting quiz. So there's one there on the structure of flowering plants. There's 10 particular questions. You just have to go through each one and you'll be given a score right at the end. We also have PowerPoint presentations, which you can click into for some extra notes. And then we do have our resource packs as well, which is a bundle of H1 notes, which you can either look at and take notes down, or you can save them and print them off. So we can see this is the ones associated with today's content. Everything you need to know about the structure of flowering plants, xylem, phloem, everything else in between. So make sure, head to our ExamRevision website. You will find out all of the information about the subscription models, how much it is per month, and you'll also find out all of the information about any revision courses which we're running. So these generally run at all of the midterm break, so February midterm break, at Easter holidays, and then also just in May, start of June in the run up to the exams as well. Do keep an eye on our social media for all the upcoming Saturday sessions as well. And thanks a million for coming to today's one for Leaving Cert Biology, and I hope to see you again soon.